Very good. All right then. Again, welcome everybody. Um, we're going to tag team this, so I will invite Mrs. Clark to chat with you for a little while.
Hi, I want to welcome everybody to the, first of all, to Governor's School, all of you as rising juniors, but specifically I want to welcome you to the Biological Sciences Strand. It is a 2-year course, so you'll take chemistry next year as a junior, and then as a senior, you will take biology with Dr. Patterson. Both of the courses are dual enrolled, and you will be registering for those both in the fall and in the spring. And if you have any questions at all, make sure you put them in the chat, and then we'll add them to that document, the Q&A, at the end. Also, I encourage you that if you have specific questions that pop up, you can always shoot me an email, and I'll be happy to answer those questions if they're more specific to your course for next year. Okay, you want to advance the slide? Okay, again, my name is Jennifer Clark, and I've been teaching this course at the Governor's School for a long time. So I've been here for quite a few years. And go ahead and advance to the next slide. And the course is a very traditional college level chemistry course. It's the same course that students would take if they were taking freshman chemistry, for example, at Virginia Tech. The topics are all listed there for you, and there's a picture of our textbook, which is the chemistry book by Zumdaw. Many of the topics we'll cover in the fall are going to include things like stoichiometry, acids and bases will be covered, as well as gas laws, atomic structure, those sorts of things. So first semester, we will really dip into a lot of what you learned in your first year courses that you're taking this year. So you'll kind of delve in a little bit deeper into those topics. And then in the spring, we'll hit more of the more advanced topics, things like equilibrium, and we'll also focus on electrochemistry. And students really do enjoy the chapter that we also spend on organic chemistry, where we just introduce that so that when you go off to college and you will take an entire year of it, it gives you just a little bit of taste of what the course is about. So the next slide. I will provide you with a very detailed syllabus in the fall, but I want to just talk briefly about some of the things that go on within the classroom. That is a picture of my classroom. We do have very traditional tests, quizzes, problem sets, projects. Those things will contribute to your grade, but you also will spend about 50% of your time in the laboratory. And so in the laboratory, you're going to be looking at reinforcing concepts that we talked about in class. I might have you investigate a new concept that I've yet to introduce to you, or also as an assessment tool. Students learn that I can put lab questions on quizzes and tests, and you also have a lab practical that you'll be taking in the fall, and then you will also have one in the spring. And since the course is so lab intensive, I feel that it's only fair that I ask you to share with me what you know and what you've learned about the lab in the form of an assessment. And I also want to stress as well that there's really no way that I can duplicate the learning that goes on in the classroom because the course is so lab intensive. And so it's really important that students attend the classes, that they're in person, because otherwise you're going to find that a significant amount of information is covered and you potentially might fall behind. We do meet our science students for 95 minutes every day, and it's amazing how much ground we can cover in 95 minutes. Okay, next slide. Here are some students working in lab. You'll see them titrating, using the spectrophotometers, using all of the various lab equipment. They really do a wonderful job of collecting the data, analyzing the data, drawing conclusions from the data that they've collected. Okay, next slide. We also work on some additional things in class as well. Students will work in groups on practice problems. Oftentimes we're using flashcard type activities to sort of reinforce concepts. And sometimes they're using whiteboards, working together. And you'll see in the lower couple lower pictures are the current students. Well, they're not current anymore. They're now our rising seniors, but they're my students from this year. And every year we have a tradition of that we tie-dye t-shirts one day, and then they all wear them on a particular day. So they are all now Dr. Patterson students. They're no longer mine. Okay, next slide. And then here's some additional activities. Usually around the winter break, we'll grow crystals and make little decorations. And then we also have a long-term lab that the students work on where they will create a coordination compound, these green crystals that they grow, and then they'll go through a process of analyzing them. So next slide. We do use quite a bit of technology as far as the interface box that works with all of our probeware. So our temperature probes, pH probes, we have melting point apparatus that we use. Also our pressure sensors, all of that will interface with the computer. And I've already mentioned the spectrophotometers that we use. And quite often I'll get a question from students or parents and they'll ask, well, what's the real difference between this course? Why don't I just take AP chemistry at my home school? And really it is the lab component. And also we don't have just one spectrophotometer, we have 10 of them. We have an entire class set. So groups of 2 can work on all of these projects. So nobody is sitting watching me do the experiment for them. They're actually hands-on working on the experiment themselves. We do, of course, utilize PowerSchool and Canvas, which all the students have become very proficient at using. And parents will have login information for both of those as well, so they can keep track of what their students' grades are. We do have a student academic support program that's in place so that if a student starts to fall behind, then we can put up the safety net and make sure that they are making the progress that they need to make in these courses. Okay.
Thank you, Mrs. Clark. So I am Dr. Patterson, and I have been at the Governor's School since 2011. I started off teaching the junior research class and have been across the hall teaching biology since 2014. I'm originally from the Midwest. And I bring my background with me to apply to our senior class in biology. As you already heard about with the chemistry class, the biology class also follows the structure of a typical college introductory biology class. It'd be the equivalent of biology 101, 102. I saw some comments earlier asking about what is this dual enrollment, what are the advantages? You will have this exposure whether you choose to dual enroll or not, which means that if you end up taking the biology sequence in college, it will be a second run through. And biology is not one of those things that you typically just grasp immediately on the first run through. A second time, third time, fourth time is how the process goes, and all of the biology classes will build upon that. Those of you who do decide to dual enroll, you will have the basis, biology 101 and 102, and will be well prepared to move on into the next class in the sequence, whatever that class might be. It's often something on the order of cell biology. We use this text by Campbell. It's quite a meaty text, and so we supplement it with a lot of videos, with tutorials to make the information much more accessible. And then, of course, a lot of laboratory. To keep things pretty consistent, now that you will have had a very good introduction in chemistry and you'll be used to the type of assessments that were done in that class, our assessments will be very similar as well, with a lot of pre-laboratory assignments and laboratory reports, projects and assignments to help cover the information and apply it, quizzes and tests, and we use a flipped classroom procedure, so I don't do a lot of lecturing in class, but there's lots of opportunities for reiteration, for review, and for applying the information. As you can see, we also have lots of equipment and lots of apparatus so that our lab groups are seldom larger than 2 or 3 individuals for any given activity. And our laboratories will begin with biochemistry and the application of that to understanding enzyme function. And then we'll move on into cellular structure and function. And then we start applying that information. We do a fair amount with DNA analysis and have the apparatus for both PCR and for DNA isolation and gel electrophoresis. So in the last couple of years, we've been able to add CRISPR to the different methodologies that students are learning about. We think it's really important that students are actually analyzing data, interpreting it, sharing it, because we all know that if you can't share the information, it's just like you just did it in the closet and nobody will ever know. And then applying the information. A lot of times our students will learn a methodology in laboratory and then they will design their own experimentation to follow it up. In these pictures, you can see the array of types of studies that our students will do, from streaking bacteria and doing gram staining, looking using the compound microscopes. And we have a set of 20 of those. So every individual will get lots of exposure to the compound microscopy. Dissection, you can see right up above the word analysis, there's a student who is sorting fruit flies using a dissecting microscope. So lots of different types of opportunities, no matter what you think your interests might be. Collaboration is key. And so within the constraints of understanding what good collaboration practice is, we will practice those things, including verbal explanation, collaborating to model concepts. My students are seldom very, very quiet, and we tend to try to ask the question, what would that look like? If we have this enzyme interacting with this substrate and the conditions are such as this, what would it look like? What would it look like if I had a different regulator in there? And so you see a lot of discussion going along in my classroom. We also use Canvas heavily, and that is one vector by which we can offer opportunities for students who have to miss a class, and that will happen to everybody at some point. Maybe you have a conflict with an athletic event that has kept you away extra long time, or maybe you're ill, or any number of things. And so we have been having live class links with our students to participate. And it's not at all infrequent that when we have our small laboratory groups, that the person who is away is actually on a personal device and FaceTiming our students so that they can see what's going on. So in these pictures, you can see up there next to the word Canvas, the thermocycler, so that we can do PCR, again, dissection, electrophoresis of DNA gels, just underneath the thermocycler. Dissection down at the bottom, those are 3 hearts. This year, the students dissected a frog, a shark, and a rabbit. So they're looking at the 2, the 3, and the 4 chambered hearts there. So we do a bit of comparative anatomy and physiology. So that's the end of what I have to present. I'm just going to take a look at some of the questions. And it looks to me like we will be answering most of these on our FAQs so that everybody can see the answers to those. I don't see anything that we need to deal with at this moment. And yes, textbooks are part of our curriculum, so it's not like you're going to college. This is still high school. We'll just be working with college level material. So our next opportunity for you starts at 7:30, and Dr. Ellen Fithian will be speaking. She is the STEM Innovator and Outreach Coordinator. She does a lot of work with students who are applying to colleges, and so she's going to share a bit of her wisdom. You can see at the bottom there the link, but we're going to try to put that link into the chat and make it so that you can get to it right away. Let's see if I can do that right now. I'm hoping this is it. I believe that is it posted. And so I will suggest that you take a moment and then join that next meeting. Mrs. Clark, do you have any finishing comments? No? All right, then thank you everybody for attending this evening session. And again, please don't hesitate to contact us by email. You can use our general email, which is firstname.lastname@nhrec, and you can find us on our webpage, nhrec.org. Thank you very much, everybody. Yeah, we'll see you in the fall.